thanks very much, guys, for having me here today. I firstly want to start by thanking Fatty and Marissa, the guys from BioCrates, and of course, Teodoro for inviting me here today to speak to you guys. So as Marissa says, my name is Steward Graham. I'm the Director of Metabolomics and Alzheimer's Disease Research at Beaumont Health, which is actually now Spectrum Health Beaumont Health. We're actually the largest health or the largest hospital chain, if you will, within Michigan. And I'm also an associate professor at our local school of medicine. So today I'm going to talk to you about metabolic perturbations in Parkinson's disease. Now, I don't want to get into the weeds of the science. So basically what I want to talk about today is it's basically a story, if you will, on how we apply metabolomics and the benefits of it and what we get from it. We've been looking into Parkinson's disease now in our lab, investigating our lab for, okay, I'm tall. There we go.
How's that fine?
Buddy, are we good? Okay. Gotta make sure everybody sees me. As I was saying to the guys last night, this is a face for radio, but we'll not tangentialize. So yeah, so we've been working with the biocrates kits and so forth for quite a few years now. We've been investigating Parkinson's disease, and it's been taking us down a couple of different rabbit holes, but we've latched onto something now which is it's quite strong, which we believe is strong, and it does resonate with Parkinson's disease and not just correlate with it. So I just want to give you, what we've been doing the last few years and where we intend to go and the benefits of using these kits and so forth for it. So, of course, with anything, we've got a few disclaimers. Oh, snooze. Snooze. Oh. We've got 20. I just can't even get this thing away.
It won't let you just drag it down to the bottom.
Okay. There we go. Just ignore. So yeah, so my research is funded by the NIH, in particular the NIA, NINS, and NIEHS. And I currently receive funding from the Michael J. Fox Foundation. And as an older disclaimer, I consult for Coleman, Roche, and Biogen. Oh, and another disclaimer, BioCrates are not paying me to do this presentation today. So if you see a lot of BioCrates kits and everything, this isn't an advertisement for them. This does not work. Okay, so just as a simple roadmap today, I'm going to give you a little bit of an introduction into Parkinson's disease. Bile acids, which is one of the main things that we're really interested in. That's where the rabbit holes we started to go down. Why bile acids in Parkinson's disease? Our next steps, and of course our acknowledgements, because I couldn't do this without my guys. So Parkinson's disease, what is it? So it is actually the second most prevalent disease following Alzheimer's disease. And unfortunately, there's no objective tested biomarker for it presently. And I say presently because we'll delve a little bit into that later. The rate of misdiagnosis can be high because there is a lot of crossover between it and dementia with Lewy bodies because of the makeup of the Lewy neurites and the Lewy bodies and so forth, and alpha-synuclein. And it's estimated to affect 1 in every 100 people over the age of 60 at the moment. So presently, 60,000 people are diagnosed with Parkinson's in the US annually, and there's 500,000 who live with the disease in the United States, and a guesstimation of between 7 and 10 million people live with it worldwide. But so nobody actually knows how Parkinson's begins, but it is believed to be a combination of both genetic and environmental. And then some of the genetics, so we've got LARC2, which is our leucine-rich repeat kinase, which affects about 2% of people with Parkinson's disease. We have SNCA, which is the which goes to the alpha-synuclein. So hyperphosphorylated alpha-synuclein forms these fibrils and eventually forms these Lewy bodies, which are the hallmarks of the disease. We have GBA. Again, it's involved in debris clearance, if you will, within the cells. So abnormal GBA leads to non-clearance of these debris within the cells, which leads to the neurodegeneration. And then we have Parkin, or PRKM, which encodes for one of the largest proteins within the human body, which is Parkin. Again, it leads to the clearance of debris within the brain. So all of those combined probably accounts for about 10% of people with Parkinson's disease. The other 90% are idiopathic. We don't know what is causing it. And then, of course, there's the environmental factors, MPTP, which is quite a famous one. There's a Professor Langston from California who in 1982 came across a group of individuals in their 20s who were presenting with Parkinson's-like symptoms. He wrote a really interesting book in the 90s. It's called The Case of the Frozen Addicts. Basically, MTPT is a synthetic opioid. So these guys were using, what was it they were using? They were using heroin, which was contaminated with MTPT. So once MTPT gets into your body, it produces and it's metabolized to MPP plus. MPP plus is neurotoxic and it breaks down your substantia nigra, which eventually leads to the symptoms associated with Parkinson's disease. So this is the first recorded case of environmental factors of Parkinson's disease. And we actually use it now as well for developing mouse models and so forth by injecting them with MPTP. So a lot of this information is taken from the CDC, the Parkinson's Association, and of course, Michael J. Fox Foundation, if you want to look into it anymore. So this is probably what a lot of people associate with Parkinson's disease. We have the tremor, so you can see it in most of the extremities, your hands, arms, legs, jaws, and face. The rigidity, where you're going to have the stiffness of the limbs and the trunk, the postural instability, and the bradykinesia, or the slowness of movement. So these are probably the majority of symptoms which people associate with Parkinson's disease, but there are other symptoms as well, which we'll talk a little bit about. I'll just touch on them. So the Parkinson's disease pathology within the brain. So we know, well, I just talked about it. We have Lewy bodies and Lewy neurites, so they make up the hallmarks of the disease, which you can see here, the alpha-synuclein, which is misfolded or hyperphosphorylated to produce these. And then the second, the second prominent cause of Parkinson's disease within the brain is the loss of these, if I can use this little, you can see it, is the loss of these midbrain dopamine neurons. So it's the breakdown of these within the brain, and also the breakdown of substantia nigra. So what's happening is that there's no dopamine being released at the end terminals within the striatum, which leads to motor symptoms associated with the disease. But it also involves a lot of other non-motor related symptoms before the motor symptoms become apparent. So you have hyposmia, you have constipation, you have depression. There's a lot of other things which go into it in the pre-symptomatic stages before the clinical symptoms, such as the motor symptoms, become apparent. So hypobrac, you can't actually see it there because this lovely little thing's covering it at the bottom. Let's see if I can move it away. Oh, I can't. So Heiko Brack, basically, he's a neurologist who defined Parkinson's disease pathology across the brain, and he believes that it starts in 6 different stages. The first stage, we're going to see it in the dorsal nucleus or the olfactory bulb. So like I said, we got hyposmia, constipation, bladder disorder. So hyposmia, for those of you who don't know, is the reduction in your ability to smell. So one of the things at the minute as well, and maybe I'm tangentializing again, is that we're very interested in this because one of the things that happens when you've got COVID is you've got hyposmia or anosmia and agusmia. So you're losing your sense of smell and your sense of taste. So there's a lot of people who are hypothesizing at the minute that those people with long-term COVID could end up or see an uptick in people with Parkinson's disease because the symptoms, the way it starts, the exact same. Again, it's a little bit interesting for me. And these again are in the pre-symptomatic stages. In the second case, we see it move to the locus coeruleus. And again, you get sleep disorder and depression. At stage 3, this one starts to hit the substantia nigra and the amygdala. So this one, the breakdown starts to happen of these dopamine producing neurons, which are projecting in the striatum. And then stage 4, it's moving again. We have the mesocortex. And then stage 5 and stage 6 is when you're going to consider it as manifest Parkinson's disease. And that's when you're going to see those hallmarks or those common symptoms associated, those motor symptoms associated with it. So one of the really interesting things was that Brack and his group, again, they hypothesized that it starts by 2 ways. Parkinson's, it either starts through the nose and the olfactory system, or it comes from the gut. And this is one of the things that we've been concentrating on in terms of, in particular, the gut. So that actually says bile acids. They're apathetic end products of cholesterol. So apathetic, for those of you who don't know, they have both hydrophilic and hydrophobic part to their molecular makeup. They're produced in the liver and subsequently secreted to the gut, where they are glycine or taurine conjugated by the microbiota. Their primary function, so is to aid digestion and the absorption of these dietary lipids and fat-soluble lipids, or vitamins, sorry. And their secondary function, they act as steroid hormones, steroid hormones, where they modulate various cell signaling cascades by the activation of membrane-bound receptors, which is something again which we'll touch a little bit further on as we go on down the line. While they are secreted into the gut, 90%, about 90%, are reabsorbed into the portal system via the enterohepatic circulatory pathway and recycled in hepatocytes. However, disruption to this reuptake leads to the spillover of the bile acids into the circulation, and this has pathological effects, in particular the neuropathological effects via the enteric nervous system. So again, we're going through the gut and we're getting to the brain. So this is a sexy term at the minute. This is the gut-brain axis, which leads us on then to some of the work that we've been doing. So a good colleague of mine, Patrick Brunden, who was previously at Van Andel, which is a research institute within Michigan. He's now one of their leaders at Roche for movement disorders. His group developed this really, really interesting prodromal mouse model of Parkinson's disease. It was the first one ever to be done. I didn't even know that he was an hour north of us, but I gave him a call. I explained to him, I'm really interested in what your mouse model does. I would be very interested in using it. I think we could identify biomarkers. There's a lot of stuff that we could do. We could investigate the etiology, the pathophysiology of the disease. And he says, absolutely no problem. 3 months later, he sent me all the mice brains. He sent me all the bloods. He sent me absolutely everything. And the other thing why he did it as well, because he's a big Manchester United fan like myself. So we got on really, really well. So this was our first study. So we used the biocrates kit again. It was the P180 or the older kit, which is available. We combined that with our TQS to profile the brain. And then we also used NMR. So the kits will give you a certain number of metabolites and so forth. But NMR is completely complementary because it's absolutely quantitative as well, and it's so robust. I could run a sample on my NMR tomorrow and send it to Australia the next day, and it's going to give me the exact same result. So it's 99.99999% reproducible. So we use these technologies to increase our coverage of the metabolome. So this thing is in my way too. Let me see if I can move this. There we go. So one of the things that we did, like I said, I'm not going to really get into the weeds today about, okay, I can start showing you mass spectrums, I can start showing you chromatograms, I can start showing you all these different things, but I think the story is more important. One of the things that we found from our brain metabolite set enrichment analysis, or MSEA, was bile acid synthesis. And I haven't circled it here, but the thing right above it as well, which is taurine and hypotaurine metabolism. So taurine becomes important when you think about when they're secreted from the liver into the gut, they become taurine conjugated. So we found that those are the top 2 metabolomic pathways which are affected within the brain of this prodromal mouse model. This actually led us on to, okay, if we're seeing this in the brain, we couldn't see it in the blood from the analysis that we did in the first instance because we weren't really measuring bile acids. We were doing it in a roundabout way, if you will. But what we decided was, are we seeing these changes in bile acid metabolism being recapitulated within the blood of said mouse model as well? And what we decided to do is that we kept some of the mouse blood back and we analyzed it this time using the BioCrates bile acid kit. Like I said at the beginning, this isn't an advertisement for BioCrates. This is just some of the ways that we got the answers for the questions that we were asking. And interestingly, the serum bile acid levels, we were seeing a 14-fold decrease. So here in the bottom, I'll just explain it, our human monomers. So these are our controls and our preformed fibrils. These are our preformed fibrils of alpha-synuclein, which we inject into the brain, and then we see the progression or the pathogenesis of the disease over a certain number of months. So we've seen MCAO, which was at a lower level in our mouse model as compared to our controls, but you only see this in mice. You don't see MCAO in humans, but we do see the taurine conjugated form of ursodeoxycholic acid. And then again, we've seen ursodeoxycholic acid both at 17 and 14 times reduced levels within the brain. And what I can say is that we actually produced biomarker panels based on this information as well. And we were looking at like 95% accuracy, or an AUC of about 95% with about 90% accuracy and sensitivity and specificity in around the 90% range as well, following tenfold cross-validation. So it was a turn up for the books. And it was one of the things that led us to getting funding from the Michael J. Fox Foundation. So a very, very interesting finding. So again, we're looking at it, we can see, okay, we can see it in the brain, we can see it in the blood of this model. Bile acid metabolism is absolutely affected. So one of the things that we did was, okay, so let's just have a look at it in terms of the pathway. Now, this is a condensed version of bile acid metabolism. It's actually quite complex, but what we found was that it actually prefers the alternative pathway on our Parkinson's disease mice. So the ones that are highlighted in blue are what are considered neuroprotective, and the ones that are circled in red are considered neuroprotective, and the ones are neurodegenerative, if you will, so neurotoxic. So what we found was, okay, once they're secreted from the liver into the gut, our UDCA and our TUDCA, they are both down-regulated as opposed to the other ones. So our neuroprotective compounds within that given pathway at a reduced level. So it's maybe accounting for some of the things that we're seeing in terms of the Parkinson's disease progression. So we found it in our mouse model, a validated mouse model, and we said, okay, does this translate to humans? So we actually contacted one of our guys, one of our good friends, Jan Asley, who's been collecting CSF from Parkinson's disease patients with sporadic, and those patients who are genetically predisposed to Parkinson's disease for a number of years. And we said, look, is there any chance that we can get CSF from you? We've got these lovely findings. Can we look into this more? Because we want to see if it's actually being recapitulated in humans. Loved the idea, and a colleague of mine, Dr. Yilmaz, he took this one on, and again, he analyzed this using, in this instance, it was the P180 again, and the NMR. Actually, this is Ali's wheelhouse. This is his NMR, which he uses. So one of the interesting things here on the left in A, we have sporadic, and on the right, we have LARC2, those people who are genetically predisposed. But one of the things, it's not that high up in it, but again, bile acid metabolism is being highlighted as two of the important pathways as being perturbed in those individuals with Parkinson's disease, be it sporadic or genetically predisposed. So this is a really interesting study that we did then. So we found it in the CSF, so indirectly we're looking at it in the brain. It's very indirectly. But we said, okay, we know that there's an interest in the gut as well from what we've seen previously, because we know it's being secreted into the gut from the liver. But a good colleague of mine, Dr. Vivian Labrie, who sadly passed away a couple of years ago, and I got talking about this, and she did this huge meta study with a large Scandinavian cohort. Basically, they traveled to Sweden, they sat down, they looked at all the data. They're looking at hundreds of thousands of data sets, and they found that those people who had an appendectomy were at like a 20 or 30% greater or lesser chance of developing Parkinson's disease. Now, that doesn't mean that you just go out and get an appendectomy. It just is, this is one of the findings that they had. So this led us to start thinking, okay, we should really be looking at the gut. We should be looking at the ileum. We should be looking at the appendix as well in those people with Parkinson's disease. So this is one of the things that we did. Again, we're looking at bile acids. So the aim of this particular one was that, is the appendix microbiome affected in Parkinson's disease and the consequences of these microbial alterations? So we used a meta-transcriptomics analysis to study the functional microbiota in these Parkinson's disease patients. And what we highlighted was that gut dysbiosis affects, gut dysbiosis affects several metabolic pathways or metabolic pathways to include lipid metabolism. And then we also found that there's an upregulation of the bacteria responsible for the production of these secondary bile acids. Again, we're starting to corroborate, and we're starting to stack the information and the evidence in terms of bile acid metabolism of Parkinson's disease. Again, we ran a quantitative bile acid analysis, and we found that there was an increase in hydrophobic and secondary bile acids, in particular deoxycholic acid and lithocholic acid. So this is one of the things that I'm very, very interested in, is that we do metabolomics, but the problem is that we need to corroborate and validate using other methodologies as well. And one of the ways that we did it in this, we used a proteomic with the guys at Michigan State University who ran it for us, and a transcriptomic analysis of the appendix and ileum to validate our findings. Again, we highlighted the changes in the Parkinson's disease gut, and they are consistent in terms of bile acid control. So we found alterations in mediators of cholesterol homeostasis and lipid metabolism. But the major finding, the major takeaway note from this was that the microbially derived toxic bile acids are heightened in Parkinson's disease, which suggest biliary abnormalities, which may play a role in the pathogenesis of Parkinson's disease. So We found that there's a decrease in our mice and so forth in those neuroprotective bile acids, but then in the humans and in their gut, we're finding an increase as well in those compounds which are considered neurotoxic. So this has led on to our recent manuscript, which has just been published last month, where we're integrating metabolomics and epigenetics to study post-mortem human brain of those individuals affected by Parkinson's disease. So we've shown it in CSF, we've shown it in the gut, we've shown it in mouse models, we found it in mouse model blood. So again, in this instance, we used the Illumina. It's an epigenetic kit. Sangeeta, who's probably online, will be able to explain it a lot better. She's an epigeneticist. But we combined it with what's called the new Quant 500. And again, with our NMR, you'll see that we combine NMR and mass spectrometry all the time. Again, not trying to get too much into the weeds here because I could spend an hour on this manuscript alone, is that the major thing that we found out on the bottom right-hand side was primary bile acid biosynthesis was the major biochemical cascade which was perturbed in those people who are suffering from Parkinson's disease. And that wasn't just from using metabolomics analysis. We also found that using our epigenetics analysis. So in the first instance, we did one, we did the other, and then we did the combination of the two. So our findings do support that the hypothesis that interactions between the metabolome and epigenome do have a significant impact on the pathogenesis of Parkinson's disease. Those of you who maybe aren't familiar with epigenetics, basically we're not studying changes to the genome, we're basically changing how those genes are turned on and off. So what we did in this study is basically we did our metabolite set enrichment analysis with our metabolomics data, and we mapped those epigenetic changes onto those specific pathways. And then we were saying that the enzymes responsible for the conversion of one to the next in that biochemical cascade were perturbed. So it was an indirect way, if you want, of looking at the proteomics of it all, or the transcriptomics of it all as well. But it has its limitations. The accurate role of these interactions are unclear. That much is obvious, but we need to do more functional studies to try to understand these. So that brings us on to where we are going now in terms of our research. So we're interested in measuring the brain lipidome because of what we're seeing in the gut dysbiosis within humans. So we're very, very interested in doing that. Are these changes recapitulated in other tissues? And can these bile acids be used as potential blood-based biomarkers? But what I can say is that these three are done. We have measured the entire PD brain lipidome using the lipidizer. So we're looking at about 1,200 different lipids at about 15 different classes. The results are very, very good. But one of the problems is it's very, very difficult to do the pathway analysis when you've just got lipids. There are some things out there which will enable you to do it, but we found that it's very, very iterative. Are these changes recapitulated in other tissues? Yes. We've been working with the Michael J. Fox Foundation. We've been using their LARC2 cohort consortium. We've mapped these using metabolomics. And can bile acids be used as potential blood-based biomarkers for Parkinson's disease? That manuscript will be long coming out. It's sitting with commercialization at the moment, so I'm not going to talk too much more about that. And can we measure the entire bile acid biochemical cascade in Parkinson's disease? We're hoping that we can. It's making up part of our next grant. And then, of course, we're working now with our mouse models and induced pluripotent stem cells as well to do more translational based studies if we can identify potential therapeutic targets and just try to understand what's happening in terms of flux-based analysis and so forth with bile acid metabolism. So that's actually making up our R01 application, which is due now in October. So acknowledgements, I have to acknowledge my guys. They're all online here today, I'm guessing. So I couldn't do this without these guys. They're absolutely fantastic. They come to the lab every day. There's a lot of this stuff just comes out of my brain when I'm sleeping at night, and then these guys make it come true. So yeah, Ali, Nadia, Nazia, and Sangeeta, who are online there today, they've been absolutely fantastic. Of course, Dr. Bahadur Singh, who is my department chair. It sounds weird that I'm in the department of OBGYN, even though I'm a neuroscientist, but he's been the guy who's been forefront in supporting our research all along. And then, of course, there's Tammy Ostentowski, who's my research nurse, clinical research manager. She basically runs all of our collections. So we do a lot of biobanking from Parkinson's disease patients. We do all the in-house cognitive testing. We're doing a lot of that and with our Alzheimer's disease patients as well. And she's absolutely fabulous. She runs a couple of other nurses as well, or manages, sorry, a couple of other nurses as well here in Berlin. I had to mention our maternal fetal medicine fellows, because these guys dealt with all the poop. and it's not nice, but they're very, very good at it, and they're happy to do it. So I said I have to mention them in this. And then, of course, our clinicians who have been phenomenal in terms of support. We have Dr. Adam Coleman, who's our movement specialist and has helped set up a lot of these collections which we're doing. Dr. Imam and Dr. Maddens, who have been supportive of me since day one. So the past 8 years, Dr. Maddens and Dr. Imam, they found money to help me go and do these studies. So they've been absolutely fantastic. So on that note, oh, sorry, I also have to thank, there's Dr. Patrick Brendan, who has been absolutely brilliant. He's been an ear for all my complaints and things which have gone wrong and helped me, discover different things. And then, of course, Vivian, who recently passed away as well. And then there's our funding bodies. And then with respect to this one, it's the majority of it comes from the Michael J. Fox Foundation and the Fred and Barbara Family Foundation. So thank you, and I'm happy to take any questions.